Hello class. Last time we have learned about the bacterial cytoplasm. In this lecture, we are going to learn about the bacterial endospores. Endospores, dormant cells formed within a so-called mother cell, are fascinating bacterial structures only produced by certain members of the genera Bacillus, Clostridium, and Sporocercina within the phylum Firmicutes. Electron microscopy has been an important tool for dissecting endospore structure. Electron micrographs show that the spore consists of a core surrounded by inner membrane, germ cell wall, cortex, outer membrane coat, and extrasporea. The core has normal cell structures such as ribosomes and a nucleoid, but has very low water content. The core is surrounded by an inner membrane which is in turn covered by the germ cell wall. Next is the cortex, which may occupy as much as half the spore volume. It is made of peptidoglycan that is less cross-linked than that in vegetative cells. The cortex is surrounded by a phospholipid bilayer called the outer membrane. Outside of the outer membrane is the coat. It is composed of more than 50 different proteins, which are highly cross-linked to each other. Finally, many endospore-forming bacteria produce spores enclosed by a thin, delicate covering called the extrasporea. Bacterial endospores are the hardest of all life forms, capable of withstanding extremes in heat, drying, freezing, radiation, and chemicals that would readily kill ordinary cells. The ability of the spore to survive heat, radiation, and damaging chemicals requires that its enzymes and DNA be protected. The spore coat protects the spore from chemicals and various lytic enzymes such as lysozyme. The inner membrane is extremely impermeable to various chemicals, including those that cause DNA damage. The core has very low water content, high amounts of dipicholinic acid complex with calcium ions, and a slightly lower pH, all of which contribute to spores' resistance to harsh conditions. As shown in the figure, sporulation may be divided into 7 stages. The cell's DNA is replicated, stage 1, followed by an inward folding of the cell membrane to enclose part of the DNA and produce the full spore septum, stage 2. The mother cell membrane continues to grow and engulfs the immature endospore in a second membrane, stage 3. Next, cortex is laid down in space between the two membranes, and both calcium and dipicholinic acid are accumulated, stage 4. Protein coats are formed around the cortex, stage 5, and the maturation of endospores occurs, stage 6. Finally, lytic enzymes destroy the sporegum, releasing the spore, stage 7. Sporulation requires about 10 hours in Bacillus megaritum. The mature endospore is located in a characteristic location in the mother cell, depending on the species of bacteria. Endospores may be centrally located, close to one end, or terminal. The transformation of dormant spores into active vegetative cells is almost as complex as sporulation. It occurs in 3 stages: activation, germination, and outgrowth. Activation is a process that prepares spores for germination and can result from treatments such as heating. This is followed by germination, the breaking of the spore's dormant state. It begins when proteins called germinant receptors, located in the inner membrane, detect small molecules, such as sugars and amino acids. Upon detection of these molecules, a series of events occur, including release of the calcium and the dipicholinic acid complex, breakdown of the peptidoglycan in the cortex, and water uptake. Eventually, water levels inside the germinating spore reach those characteristic of vegetative cells, and enzymes in the core become active. This allows the spore to begin synthesizing the various molecules it needed to initiate spore outgrowth and return to a vegetative state. Okay, that's all for today's lesson.